Hi everyone, today I'm going to be covering different types of cell movement, which is an important topic in developmental biology. There are 5 main types of cell movements in gastrulation, and these are invagination, involution, ingression, delamination, and epibole. And these 5 cell movements in some combination form all gastrulation events. We're going to start off by covering invagination. To represent this visually, I'm going to start off with a little blastula with no differentiated germ layers. And so this motion is kind of like the forming of a pocket that is formed by poking inwards. You can think of this motion kind of like poking your finger into a balloon. And this motion will continue to form a larger pocket. And in this case, we are going to be using the example of sea urchin gastrulation, and so here this invagination leads to the differentiation of the endoderm and the ectoderm. So that's invagination. Next we're going to be covering involution. Here again we're going to start with a little blastula, and a pocket will start to form in the blastula. At first this seems a lot like invagination, but it's actually different because of the movement of the cells. Here, the cell movement is kind of rolling inwards, and you can think of this rolling motion like the motion of a tank's treads, how it kind of moves over one another. And this type of cell movement will continue to form the pocket, which will get larger. And here we are using an example of amphibian gastrulation, and in that case, this involution will lead to the differentiation of the 3 germ layers, the endoderm, the mesoderm, and the ectoderm. So next we're going to cover ingression. Again, here we're going to start with a little blastula, and here individual cells will disassociate from the ones beside them, becoming mesenchymal cells. Then these mesenchymal cells will migrate upwards, but they will move independently from one another, which is important because this factor helps to differentiate it from the next type of cell movement that we're covering, which is delamination. In delamination, we're again going to start with a little blastula, and then some cells will dissociate from their original layer. However, this time these cells will move inwards together to form a new layer. And here, unlike ingression, the cells that move inwards to form the new layer are epithelial. So in this example, we have 2 layers as the result. However, there are cases of delamination where more layers are formed, which is important to keep in mind. Next, we're going to move on to our last type of cell movement that we're covering, which is epibole. Here we're actually going to start later on in gastrulation for our example. So you can see we already have 3 germ layers differentiated. In the middle we have the endoderm, then the mesoderm, and then those layers are wearing the ectoderm, which is in blue, kind of like a hat. And in this type of cell movement, which is pretty simple, we'll have this ectoderm move to cover the rest of these cell layers, kind of spreading over them. And you can think of this type of cell movement kind of like pouring chocolate syrup over a scoop of ice cream. So here the chocolate syrup will kind of spread out over it until it covers the whole scoop. So to recap, we have just covered 5 types of cell movement which are important in gastrulation, and these are invagination, involution, ingression, delamination, and epibole. Invagination, we form a pocket kind of by poking a layer of cells inwards, like how a balloon would react if you poked it with your finger. Next, we have involution, which forms a pocket by kind of rolling cells inwards, like a tank's treads rolling over each other. Then we had ingression, where mesenchymal cells disassociate and move inwards independently from one another. And then we had delamination, where epithelial cells move inwards together to form 1 or more new layers. And then finally, epibole, where an epithelial sheet of cells move together to envelop deeper layers, kind of like chocolate coating that spreads over a scoop of ice cream. So those are our 5 types of cell movements that we're covering today. Thank you for watching.